Hi guys, last time we were talking about the chemical transformation of food and we said that there is something in the saliva that is transforming the carbohydrates or the starch in the bread into reducing sugar. And we showed that by performing the iodine test, which showed that in the presence of the saliva, first when you have carbohydrates it gives dark color, and then in the tube where saliva is present the Fehling test gave brick red precipitate. However, in the absence of saliva there was no brick red precipitate, indicating the absence of sugar. Thus the saliva transforms the carbohydrates into reducing sugar. And I asked you to solve all the parts, and we still have part K, which is to schematize, means to draw with labeling the biological phenomena that occurs in tube A. So you have to draw the carbohydrates, which are a long sequence of sugar, and in the presence of the saliva, so you have saliva, it's transformed into reducing sugar, which is two sugars. So you have lots of sugar here and it's transformed into two sugars. So you have polysaccharide transformed into disaccharide. This is the schema, and do not forget the title. So what we are going to talk about today is that there is something called enzyme, which is a chemical substance that is present in the saliva. Okay, so here you have in the saliva there is a molecule called salivary amylase, which digests starch in the presence of water into maltose, which is the reducing sugar. Okay, in neutral medium, you know, we have acidic medium, basic medium, and the neutral medium, which has pH equals 7. This is called molecular simplification, where we are simplifying huge or large molecules into smaller ones. So this salivary amylase is called enzyme, and we have different enzymes in our digestive system present in the digestive juices. You have the amylase, you have the pepsin, the trypsin, you have the lipase and maltase. So these are the things that we are going to talk about in our new lesson, and mainly the conditions for these enzymes to act properly. Okay, so I will ask you please to open on page 158, and we are going to start with enzymes, agents of digestion. So here you have Sami, who is a student in grade 9, who asked his teacher, can one enzyme digest different organic matter? So let's say that you have the salivary amylase. Can salivary amylase, we saw that it digests carbohydrates. However, can it digest proteins? Can it digest lipids? So this is what we want to know. Can one enzyme act on different substrates, different food, or each enzyme is specific for certain substrates? His friend Ali jumped and answered, I think that every enzyme is specific to only one substrate. So this is our hypothesis. This is the objective of the experiment. We want to see if each enzyme is specific to only one substrate. So you have here cooked fresh saliva, cooked starch with fresh saliva, and you have albumin of an egg, which is a protein, and also with fresh saliva. The saliva, as we said, it contains salivary amylase. So